Hey, good morning, everybody. So you heard a lot about the progress that we have been making on the wastewater side. And I would say, like, maybe what I'm talking about today will not be necessary in the future once we have solved that problem. But until then, we still have another problem, which is sometimes considered the legacy nitrogen, the nitrogen that has already entered the groundwater. And with the hydrogeological characteristics of Long Island, we have opportunity to deal with this with permeable reactive barriers. You see here two maps modeled and extrapolated from measurements, the distribution of groundwater nitrogen, both like on the east end of Long Island and then towards the southern base. You see that it's kind of patchy, but you see that in many places the concentrations are actually much higher than the 3.8 average. And so these are the sites that we may consider, or that we are considering for the installation of these subsurface systems. You saw these pictures before. The groundwater, as Long Island is a watershed, flows through slowly towards the base, and actually it's not that slowly if you consider groundwater velocities of about a foot per day. So we can use that flow, natural flow of water by installing permeable reactive barriers that then can treat that water. On the right side, you have seen probably maps like this that give you an idea about the timescales we are looking at. Depending on how close you are to the shore, it may take a year, 2 years before groundwater is entering surface water. But if we look further upstream, it may take decades to hundreds of years. So places that have been loaded with nitrogen in the past, these are places where we should pay our attention to help us until we have solved the nitrogen problem at the source. So installing PRBs relatively close to shores has a benefit that at close to the shore, the groundwater is actually, the velocities are relatively high, and we can use this velocity with only pumps, these are passive systems, just intercepting the flow and removing nitrogen. There are in principle two basic carbon sources that have been used also for other contaminants. These are wood chips, and these are oil, food-grade oils. It sounds a little awkward to put oil into the sands, into the aquifer, but these carbon sources, they release carbon very slowly, and that's exactly what we want. We want to have them in place for a long time to deal with the nitrogen through the process of denitrification. Wood chips can be inserted like in trenches. We can create column arrays. Carbon oils can be injected directly into the grounds, which has some benefits. So CCWT tries with lab studies, with column studies, with pilot systems in the field, but also already more extensive field installation to understand those systems specifically for Long Island's aquifer characteristics. And we are trying to figure out, okay, what is, for example, the optimal thickness of a PRB so that we don't overtreat the water. That's something we want to avoid, simply because if we overtreat the water, we will produce methane. So we can choose an optimal thickness of these PRBs to not overtreat the water. When it comes to column arrays, like on the bottom right side, we should consider, okay, how many rows of PRB columns, wood chips or injected oil should we have to exactly optimize the setup for site-specific conditions. So laboratory studies look like this. Early on, we ask ourselves, so what is the nitrogen removal potential of different types of wood chips? You saw figures like that from Stuart before. There are differences between wood chips. This here just gives you an idea about the nitrate removal of aged wood chips. So these are wood chips that have been in the ground for quite some time, and you can see that they continue to remove significantly amounts of nitrogen. It's temperature dependent. Different types of wood chips perform differently well, so we can consider that information when we install PRBs and make them, optimize them for these site-specific conditions. One system that was really helpful in this initial phase was a PRB test cell setup that we installed together with Ron Paulson was really the spearhead behind that, installed behind a bulkhead. That was kind of a novel idea, perforating bulkhead that needed to be replaced anyway. perforating its subsurface so that groundwater can flow through these test cells and then into the marine sediment and eventually then into the bay. And this allowed us to really scientifically address many questions related to how thick a PRB should be. Do wood chip columns do the same kind of job as trenches? And so over the years we have then, these replicated test cells. We have the ability to sample upstream, within, and downstream of these test cells to figure out how they perform. The first point I want to make is, just like over the years, we know that the nitrate that comes in is variable in time and space. You see here, just like the upstream nitrogen concentration that goes into each of these test cells, which are just like 3 meters apart. And you see like each of these points is different. That means there are areas where nitrate is a little higher than a little lower on this spatial scale. And then the arrow bars gives you an idea about the temporal variability. So the nitrogen in the groundwater is heterogeneous. And so we are interested in what does that mean if we choose a specific thickness for a PRB, for example. Well, the answer is what you see here on the right side. These are all these data that we collected from all these different test cells. You see, each row is one of these different types of test cells. On the left and left to right is just like a replication, and then in each of these small figures, you see the different sampling points over the years. In blue is what goes in. In green is what goes out. And you see in pretty much, in the control, which don't have wood chips, you don't see anything because there's no way that nitrate is removed. But in all these other test cells, you see what's going in. Blue is high, what goes out is pretty much total nitrate removal. We also did measurements on other byproducts that could be problematic, but the system really gives us confidence that these systems work well, even so if they are that close to the surface water. We then used the data from lab experiments and from our field observations, the sediment characteristics, to run reaction transport models, because in the field you can only measure so much. With these models, we have the ability to change all kinds of things and ask questions that we would like to answer. For example, how do these systems work if nitrogen is very heterogeneous and maybe dynamic? So we can change the boundary conditions in these models. So you see here different types of simulations where we looked at different conditions and with respect to their excess nitrogen, so the PRB will not handle all the nitrate. It's a situation where nitrate is actually lower, what happens then? Or even like variable flow, like with a plume of nitrate going through PRBs. And we here compare trench type setups and column arrays, and what you see at the bottom is basically what we would measure downstream, what we would expect. The first important thing here is the wood chip columns, if they are spaced correctly, they just act like a trench. And it also shows that these systems work actually well. It doesn't really matter if there's more nitrate coming in than these systems can remove, then obviously there's still like some nitrate in the effluent. But with this heterogeneous flow, it actually works quite well. The column array actually has some benefits in terms of removal of nitrate. And then we can use these models even further and just give you a little bit of a sense of what we can do with this. We can now look at the conditions for different groundwater velocities. On the right axis, we can look at different temperatures, and we can look how many rows would be needed to remove that X amount of nitrogen. So the left axis here shows you the decline of nitrate that we would expect depending on the dissolved oxygen concentration, the groundwater velocity, and the temperature that we would find at a specific site. So for a specific site, then we can make an informed decision to optimize the setup of a PRB. This gives you a little bit of an idea about the things that we have found about the wood chip-based systems. Well, it turns out that trenching is not easy. And with the project proposals that we have written, it turns out that it can be infeasible at specific sites. For a column array, you need lots of columns, which takes a long time to establish. That's why we also now start looking more into the carbon injection idea, which could also allow us to treat groundwater deeper down, because trenching and column arrays are typically limited to the upper 10, 20, 30, maybe 30 foot of groundwater. And oftentimes we find nitrogen pollution deeper down. So the first thing that we were interested in, again through column studies, how well does this injected emulsified vegetable oil help with nitrate removal? And on the upper right graph you see, well, it's really like scaling with how much you put in. But the other question we have, we are talking about permeable reactive barriers. Permeable means like water can easily flow through them. If you inject some, the wood chips are always more attractive to water, it flows easily. But if you inject something into the soil, how does it impact the permeability of the sound? May water bypass those systems or only allow treatment outside of the amended zones? And you see, like here in this first experiment, we actually found a significant decline in permeability that was concerning to us. However, we then thought a little bit more about how we have established these amendments. And if you think about, like in a field, when you have an injection well, the front of the EVO spreads quickly at the beginning and then slower and slower towards the outer side of the injection line. We can basically calculate the expected velocities, and we realized actually the velocities with which we inject oil in the field, for practical reason, or contractors do, is actually much, much higher. We are talking about velocities of 50 foot per day. That's much faster than what we did in the first experiment. So we repeated our experiments with more realistic injection protocols. And luckily, or we are really happy to see that this negative impact on permeability was not visible anymore. In the upper side graph, you see column permeability before and after the injection. You actually can see that the permeability increased in our experiments with the injection, may it be with or without EVO. So that's really good news, but which means like if this EVO is injected at high rates, we don't create these low permeability zones. At the same time, we are now able to see where this EVO ends up and what the concentrations are. That's at the bottom graph. We inject different concentrations of EVO for different durations, 2 and 4 hours, which is typically what is done by contractors in the field. And you see this clear increase in EVO, really low concentrations, just 1% of weight, which means we really create this very thin film around these sand grains that then will allow denitrification to occur in coming years. So the next step with these experiments is again like figuring out what is the nitrate removal, what are potentially byproducts that are formed, and how can we minimize those. We now work at multiple locations. Many cases now we focus on relatively small, very impaired water bodies where we know even relatively small installation could make significant improvements. One site that I'm really excited about will be Mill Pond, where we propose to basically replicate what we have done at Hampton Base behind the bulkhead, this case without a bulkhead, really like a science research approach where we have test cells. We can test carbon injections against wood chips of different arrangements. We also just received green light to start working on Wainscott Pond. On the right-hand side, you see that this is really one of the most polluted water bodies, even like in the state. Looking here at the blue and green algae concentrations. That's a site where we also found elevated nitrate concentrations between 5 and 10 milligrams per liter, relatively deeper down already. So here we propose a carbon injection approach to deal with the problem. That site will also allow us to more extensive site characterization beyond of what we plan to do. And then we have a site at Lake Agawam where the injection was just done. I'm really excited to go out later with the people and get the first set of samples. It takes some time. The groundwater velocity there is maybe half a foot per day, so it takes some time until we'll hopefully see the changes that we have done through the injection at this line with these pink dots. And we expect almost 300 pounds of nitrogen will be removed through this relatively small installation over the coming years. Lastly, I want to mention that DEC, and we work with DEC to publish a document. You can use the QR code to download it, but if you search for it, this is really meant to be a guidance document for stakeholders, for contractors to inform them about best practices on how to choose between a wood chip or injection PRB, how to come up with the best thickness of these systems so that they are not over-designed, and also thinking a little bit about the permitting processes that needs to be in place before you can start doing something like that. So with that, I would like to, well, so that's all I have to say about PRBs. Taya Clemens, she has a poster outside. If you want to know more about it, please visit her poster. And so just like I started to talk about regulations and permitting, so I want to invite Jennifer Jüngst here to the podium to talk a little bit about updates on that front. Thank you.